Hi, everyone. I'm here with Bodil Willumsen. She used to be the head of EU supplier relationship at Novartis, so she was responsible for lentiviral development for Kymriah from the very first patient treated all the way to market approval and post-approval. And then lately, she was also involved with AVV gene therapy with Luxturna and also with other consulting projects. So I invited Bodil because she's an expert in CDMO selection. And that's a very important issue that most, if not all, cell and gene therapy startups have. So, Bodil, thank you for being here.
Well, thank you. It's a pleasure. Glad that we could find a time that worked out.
Thank you. So, as I said, CDMO selection is very important. So, what is important when it comes to selecting the right CDMO? And has anything changed from when you started with Kymriah in 2013? So it's basically 10 years now.
It's a long time ago. So yes, in 2013, the cell and gene was obviously in its infancy, and there were very few CMOs that were, offering services in that field. For instance, for plasmids, there were a few, but they were not manufacturing to GMP at that time. So we had to work with generally our manufacturers to help them enhance their quality standards. For lentiviral vector, that I was also responsible for, there were two companies working with lenti basically, and there was a few academic players. So what mattered to us when we selected our CMO back then for the lentiviral vector was, first of all, their technical capabilities and their IP position. But I also would mention that what was very important was maybe the more soft skills, their openness to the collaboration, their willingness to invest upfront to show that their technology could be used for our purpose, and also their flexibility in offering a reasonable contract that was a win-win for both parties. That was very important. I mean, this was a relatively small company at the time, and we made a leap of faith to work with them, and it just worked out very, very well. So it, for me, in my work history, this is a prime example of how important it is to select the right CMO. So nowadays, the field has matured and there's a lot more CMOs in this space. And there's been a lot of talk about the limited capacity for lentiviral vector manufacturing, for AAV vector manufacturing, for instance, and also in the cell field. And so a lot of new players have come up. And some of them have a track record, maybe a good one, maybe a bad one, and some of them may not have a track record at all. So there is a lot to consider when there could also be some of these CMOs that are established in, let's say, the antibody field, and then they're just opportunistically trying to join this new field of cell and gene. And you need to think carefully before you select.
Excellent. So, what would be your recommendation to selecting one if you are if you were in a startup, in a small startup at the beginning of their journey?
Yeah,
What would be your
I
recommendation today, given that there are way more players and probably a kind of overload of information and you don't know where to start?
Yeah, I think one of the most important thing is whether you have your own process that you want to move forward with, or whether you want to rely on a platform process from a CDMO. Because if you need a platform, of course, that limits the number of CMOs that you can choose. Often it will probably be a hybrid scenario where you have some process, but it needs to be adjusted maybe to the scale and to the industrial conditions and to the quality needed for clinical. So that I think is a very important one. And then there could be a long list of things. I think that, of course, their expertise and their track record, as we just talked about. Um, as there are so many new players in, for instance, the AAV field, do you want to be the first one in that field for that CMO? Maybe not. Or maybe you're willing to give them a chance if they're otherwise the right fit, if they have a good track record in adjacent fields, maybe for other vector types and so on. That would be one thing. Then, of course, lots of other technical requirements, such as or the bioreactor type and size, the analytical capabilities, and so on. For a viral vector, there might be time savings if you select a CDMO that can also offer other services. That could be the plasmids manufacturing, or it could be the fill and finish. That might offer some time savings, and you don't have to work with so many different CMOs. You also have to consider their manufacturing capacity. How many lines do they have? How busy are they? Do you need to commit to your manufacturing slots way in advance? Sometimes you do. At least in the past, that has been my experience, that you need to lock in your manufacturing, let's say 6 or 12 months in advance. And so you want to talk to the CMO and understand what are the conditions and what is the flexibility if you need to shift your manufacturing slot. Of course, there's price. Price is such an easy thing to look at, but it should never be the overriding factor, because later on, if you want to change your manufacturing process or change to a new CMO, the cost of doing that is so much higher than maybe paying slightly more for your first batch. Yeah. Timelines, of course, how, as I said, when do you need to book your slot, how far in advance, the quality track record, the size of the CDMO. If you're a very small startup, you might not want to work with a big, rigid CMO because it might not be a good culture fit. Or you might choose to do exactly that because they know what they're doing and they have this good, well-tested platform. So finally, don't want to underestimate, as I said, the collaboration style. How willing, how flexible, and how communicative are they? These kind of soft things that you may want to go visit them before you can really truly assess. And then, of course, there can be the geographical location. But again, I wouldn't say that this is a super important one. So there's a lot to consider, probably a lot more than what I just listed, and you may want to actually liaise with a consultant who knows the field to make sure that you do the right selection up front, the best that you can.
Excellent. Well, thank you, Bodil. So now I'm thinking, still assuming you are in a startup, imagine you are in a startup with a few kind of people at the very beginning, and you might have a process, as you described, sometimes it's a bit of a hybrid, but once you selected a CDMO, maybe with the help of a consultant, what will be your recommendation in terms of tech transfer? Because then you're still a small startup, and you still need to transfer your process to the CDMO that you selected. How do you go about it?
Yeah, thanks. So you may want to consider, you probably often want to consider to do a quality audit up front. The case where you might leave that out is if you're sure that they have a very good quality track record and they've been inspected by various authorities, FDA, European authorities, whatever, then maybe you can leave it out, but you definitely want to make sure that they have a good quality system in place. Then you need to set up the team. And for me, essential is that you have a project manager on either side that function as the single point of contact into the two companies. And they will speak often, they will set the timelines and the goals, the well-defined goals, and they will set the team. And size of the team can vary a lot from 1 person to 10. I don't know if you have experts that you want to include in manufacturing, maybe quality, maybe regulatory. Then the teams need to be defined, and the experts in both companies need to speak directly to each other. And I would recommend that in email conversation that they always include the project manager on there. It's just to make sure that there is one person who has the complete overview on either side. Then I would always recommend beginning with the end in mind. And what I mean is, if your company has a research process, a process you made in your own lab, be open about it and allow time for the CDMO to test it at small scale before proceeding to full scale and to make adaptations. You may think that it works well, but it's something else to do it in routine settings and to high quality standards. So do allow time for the CDMO to do what they're good at and to adapt the process before you make your GMP batch for phase 1. And I know this can be difficult. Believe me, I know, because there's always time pressure. And traditionally, CMC, there's always this pressure from the other parts of the company to do it as quickly as possible. But my recommendation is to leave time and to make sure that you have a good process in place as much as you can. Also, you may want to consider the manufacturing line that you use if you have the luxury of choosing, because change of manufacturing line later on will require comparability studies. So it will be costly and take time. So if you're able, even if you use as a different manufacturing line afterwards that is right next to the one that you've used and uses the same single-use equipment, you still need to do comparability. That's my experience. And so if you can start in the right line that you can also use later, maybe even into commercial, but at least later on, that will be an advantage.
Yeah, avoiding having to change the process. Maybe some changes will be inevitable, but you want to reduce it as much as possible. I guess there is even more pressure when it comes to CMC in cell and gene therapy compared to other traditional biologics, because it's so much easier and so much more kind of established with other products that people underestimate that when it comes to cell therapy in particular is very challenging.
Exactly.
And sometimes it just cannot be done fast enough as you would normally do with another antibodies or another kind of traditional biologics. So I understand. And with that in mind, based on your experience, what can make the process and this tech transfer smoother and more successful long term?
Yeah, thanks. So some of the examples that I've been following over the years, so several of the CAR-T players that made it to the market have had trouble delivering enough treatments to the market after they had approval. So many have, of course, struggled to build out their cell manufacturing capacity either locally or around the globe. And this includes Novartis, of course. But there are also several examples where the lentiviral vector was the limiting factor in not being able to meet the demand. And so while there's been a lot of debate, as we discussed, there's been a lot of talk about limited manufacturing capacity for vectors, I don't think this is the issue in these specific cases. I think the issue here was that once you launch with a specific, maybe low-yielding vector process, it takes years to be able to change. You need to develop a better process. You need to go through all the same motions that you did already. You need to validate. You need to do PPQ runs, do analytical bridging, do comparability studies, including comparative stability studies. There's a big package that you need to put in place. It takes time. And then you can start your regulatory process. And in, let's say, Europe or US, you may be able to get some changes approved in half a year. There can be other countries where it takes a year. So this whole thing about changing your process after you launch is a big, demanding process that takes years. It's very lengthy. So you're really better off launching with a more mature process, if at all you can, as we discussed. So in essence, what is important is to select the right or the best CDMO that you can, with the best technical capabilities and with the most collaborative people as well. Then again, you need to allow the CDMO some time to adapt the process, to make it more robust, to make it more industrial. Keep in mind that what may have worked well in your lab may not work in an industrial setting and may not produce good GMP material for clinical studies. This is definitively also the case for cell processes such as CAR-T, that it may work well with a few highly skilled technicians in your lab, but if you go to a bigger facility with more different people, it may no longer work because it's a tricky process. If you find, as we said, the need that you need to change your process, and you probably always will, but do it as soon as feasible. Do big changes if you can before phase 2. The later you make the changes, the higher the bar will be for implementing it. And overall, as you also alluded to, CMC has a more dominant role in cell and gene therapy than in classical biologics because the biology is just so much more complicated. Of course, it's getting more and more of a commodity, but it's not there yet, right? It's not at the stage where antibodies are. So do make a point of negotiating with the project team and with your clinical and translational medicine colleagues or with your management to negotiate enough time for the CMC development upfront in the early phases of your drug.
Excellent. Well, thank you, Bodil. Thank you so much for this very interesting interview and for sharing your experience with us. I'd like to thank you, and I look forward to our workshop where we can learn and deep dive into how to select a CDMO and how to go about the tech transfer process.
Indeed. Bye. Thanks.